Come on. Hmm, I think that one. Okay, can somebody confirm that you can hear me?
Yeah, I can hear you.
Great, thank you.
Yes.
I thought I had my syllabus here. I'm not finding it. Oops, didn't want to shut that down. Now I got to go back to Clark. All right, let me share my screen. All right, you should be seeing my screen now. It looks like the lecture lesson is 74% done. My computer will be slow until that finishes. A participant has enabled closed captioning. Don't know what that message is about. All right, so today we're going to be talking about Lab 2, aseptic transfers. You do need to read the textbook on Chapter 3 throughout the section on compound light microscopy before beginning Labs 3 and 4. So you do need to read this part of the textbook before Thursday. Okay, we're going to be beginning working with the microscope on Thursday, meaning Lab 3. I also need to talk to you about your infectious disease project. You're going to begin that project today. Let me remind you to take the practice quiz 00. It has two parts, practice quiz 00A, which is short answers, and then practice quiz 00B, which is multiple choice. Any questions about what we're doing? All right, I think I will begin discussing the infectious disease project, and then we'll go on to Lab 2. Let me see if I've got Clark here. I accidentally shut this page down because it showed me Zoom and I wouldn't want Zoom. So let's talk about the infectious disease project. First, it is due by February 18th, but it will take you some time to do. You're going to do a project on an infectious disease. It is worth 100 points. This isn't what I was expecting. You do need to make it in a document, but you can take it as a docx, rich text format, and a PDF. Let me see. I've got that already. I probably do. Oh, the lecture finished. Let me finish this. Actually, I don't think it's in the lecture. I think it's in the lab. Because this is a lab project. There it is. Make sure that's the latest one. That's the latest one. All right, so you're going to be doing a project on an infectious disease. Make sure the disease you pick is infectious. If it is not, you have to pick a topic, email me to do it, and I will tell you whether or not you can do it. The first person who sends me the email will get that topic. And you cannot I'm not going to allow two people to do the same topic. Now, there are some ways to like break it up, like with influenza, for example. There are 3 different types, and one person could do influenza A, another person could do influenza B, and so you can break it up that way. But you cannot take a topic that somebody else has chosen. Why? You have to get it okayed with me, and then I will also tell you it's not an infectious disease, so pick another topic. I guess that's it. Generally, just pick an infectious disease, and unless somebody has beaten you to it, it should be okay. If you take an infectious disease, which isn't, I don't know, commonly known or isn't highlight, like the very popular ones are always tuberculosis, COVID-19, maybe the flu, the flesh rotting disease, malaria, not malaria. You know which one? Hansen's disease. Those are very popular topics. And so if you want one of those, and AIDS, you have to get them in really right away. Late in the term, you should pick a disease which isn't that popular. I guess I'll take it that way. Popularity. Like somebody had, what was that disease called? I don't remember. And that was a good one for late in the term. The idea is to make you an expert on one infectious disease. Take any infectious disease that you want and get it okayed by me, and then you can do it. You must do research about it. And while you're doing your research, you should keep list of your sources. You do need to use someplace in here. There's 4 sources that have to be good sources. And by good sources, you can use something that's scientifically appropriate and reputable, like a government website, USDA, FDA, Center for Disease Control, or their publications, a scientific journal, Nature, Science, etc., popular magazines that are known to be reliable, like Scientific American, National Geographic, Time, Newsweek, books or textbooks on biology or microbiology. A textbook is, and all of these are referenced. So any source that's referenced would be a good source. A reliable and credible newspaper may be used, like New York Times, but not the National Enquirer. A variety of scientific reputable television shows like the History Channel, PBS, etc. But be suspicious of sites that are religious, politically motivated, privately funded. These websites tend to have their own bias, and so their information may not be correct. Personal websites, anybody can put up anything on the web page. A college website that has student information, you should be very suspicious of that. Generally, college professors do not put up incorrect information, so college professors generally would be okay. Do not use Wikipedia, because anyone can go in to use Wikipedia and then change it. And I actually knew somebody who went in to a site like, I think it was Charles Darwin, and he went in and added something and it was allowed. So you can't use Wikipedia. You could use Wikipedia for your initial site, but then you got to give a source to back up any information you use from Wikipedia. So if you use Wikipedia, make sure you have another source to back it up. Usually Wikipedia will give you their sources, okay? And you need 4 sources which are reputable, okay? And where do I say exactly 4 sources? You should have a literature citation page, which gives the full citation. And there it is, 4 unique sources referenced in your report. And that'll be worth 15 points. You must use a citation style and use the same citation style for your literature citation page, the full references, as well as your in-text citations. Let me talk briefly about the in-text citation since I mentioned it. An in-text citation will be worth 10 points. In the in-text citation, you should put after a fact, which is not commonly known, the author's name and the year in parentheses. And it's usually just the author's last name and the year. And if it's 2 authors, it'll be the 2 authors' last name, like Smith and Jones, 2010, comma, 2010, and then parentheses. If it's 3 authors or more, you say the first author's last name, Smith, et al., comma, and then the year. That's how you do an index citation. Do you guys want me to write that out? Anyone want me to write that out?
Yes, that would be nice.
So in-text citation. After a fact which is not commonly known, like if you're doing something like on COVID-19, and you say how many people have died in the world, I don't know that number anymore. So you would have to give the number and then give a source citation. And we're just going to go Smith. This would be obviously 2023. Okay. And if it's more than 1 person, but only 2, it would be something like Smith and Jones, 2023. If it's 3 people, you then, for the in-text citation, go like this. You can use abbreviation for the in-text citation if it's 3 or more people. Now, in the full citation, you have to give, generally speaking, all the authors' names, and usually you give their last name and then their initials. Okay, so you give all the authors' names. Don't ask me why I gave you mine, but I just did. And then you would give the year. You also have to give, if it's a publication, you have to give the article name and where this is from. Now, if it is a scientific journal, you would give the journal name, which go Scientific American. I should have done something that was easier to, I'm going to abbreviate that. You shouldn't abbreviate it, but I'm going to abbreviate it because I'm not going to type all of it. Scientific American, and then the volume number, we're just going to put a number, and then the page numbers. Is that everything? I think that's right. The year, the name, the volume number, and then the page numbers. If it is a website, you would then give all the authors' names, the article name, which would be generally the name of the web page, and then the web page address. There are some optional things you can give here, and I don't remember how that's abbreviated, but it's some symbols. If it's something that could change on a web page, you should also give the date accessed. I might not spell that right. So if it's something that could change, like I gave you the example of the total death rate, you should put the date that you access that information because that number will generally increase. All right, if it's a book, you give all the authors' names, the article name, the book name, the book editor, the book publisher, and that's usually a company, and then the city that it's published in. Nowadays, they're published in many cities, in which case you just pick the first city that it lists, or you can pick whatever city you want, as long as it's one of the ones. Now, I don't have the year. Yeah, I do. Name. I think that's right. All right, any question about any of that?
Is this MLA or APA?
It can be either you want. It can be a scientific, whatever that's called, the citation. It can be APA citation. It can be MLA citation. If you choose one, make sure you use it all the way. Okay, don't mix up your citation styles.
I have something to share if it'll be helpful.
Okay.
The college research database, like ProQuest or whatever you can access from the library database, has a button that you can push that'll make your citation for you. You just want to make sure that it's in the right style that you're choosing and it has all the required information, but it's pretty helpful.
Yeah, actually, instructors don't like that because then you guys don't ever know how to do this, but you can use that, yes. It tends to give in all that extra information, like I think they tend to give the number or their grant number or something like that. And then there's like the Library of Congress number, something like that, that you can add on there. That's optional. All right, let's move on. I think that's good enough. Let me warn you that the citations are a place where students frequently lose points because they're not citing the article correctly. They're not doing the in-text citation correctly, or they're not doing the full text citation correctly. Usually with the full text, what they're doing is they're putting in an abbreviation or they're not including all the authors. There is one time where you can abbreviate in the full citations, I almost hate to mention it, but you have to write 7 of the first authors, and then after that, you can use et al. Okay? So you have to put out 7 of the first authors. You can't do just the first one. And that's how the full citations are done. And I hate to even mention it because that'll just confuse you. It's easiest with the full citations, just to include all of the authors. And then you won't get any reduction. All right, so the full citations are worth 15 points. The index citations are worth 10 points. Generally, when I'm looking at the full text, I'll be able to tell right off the bat whether it's a good citation or not. So I don't even have to look them up. I can usually look at it and to tell. If you give a web page for your full citation, meaning web page address, make sure it's the entire web page address, because if I do check, and I'm not saying I will, but if I do and it doesn't go to the site, you would get a deduction as well. All right, let's talk about how you do the paper. You have to do the research. I already mentioned that on the topic, and then write up your report. It must be typed, single spaced, with 1-inch margins and at least 12-point font. I will have difficulty reading anything smaller than 12-point font. I do wear these glasses for a reason.
Professor,
Mhm.
do you have a certain font type that you prefer, like Times New Roman, or do you mind any other fonts?
No, I don't care what font you use, so use whatever you like.
Okay, thank you.
It must have the cover information, but I don't want a cover page. Just put the cover information like the title, the name of the disease, your name, the date, the course name. Just put that on the same page as the first page of your text for your project. Okay, so don't use a cover page. It just takes up extra space if you do. I won't print it out, so I won't be terribly mad, but you won't get full points if you don't do the cover page right. And that's only worth 1 point. The text must describe all aspects of the disease. I'll show you what you need. The text part of the article will be 60 points. An additional 15 points are for grammar, spelling, flow, and readability. You should not plagiarize. Only use direct quotes when absolutely necessary. If 50% of the document or more is put in direct quotes, you'll get a penalty, meaning you should use your own words, but you may quote if you need to. Like if you're quoting McCarthy and he says, "I shall return," you should put that in quotes and say McCarthy. That was a general in World War II, if you don't know what I'm talking about. Not that it's important. And then after the quote, you should give a source, meaning an in-text citation. All in-text citations should be followed by a full citation. Plagiarism will result in a score of 0 for the paper, so don't plagiarize. If you use more than 3 words from some other source, you should put it in quotes and say where you got it from. All right, that's it for that. The project and the text was worth 60 points. You should break it out into sections that have a heading, and one will be causative agent. If you do not have causative agent as a subheading or a heading, you will receive a penalty. Okay, meaning I want it to be certain which is which section. In the causative agent, you should talk about what the causative agent is, If it's a bacteria, a virus, or a prokaryote. If it's a bacterium, talk about what are the characteristics of the cell, what's the gram reaction, what's the cell shape, what's the growth arrangement, how the cells are arranged, what's the metabolic capabilities, etc. What's ever important for that bacteria causing the disease. You'll have other information for a virus and a prion or eukaryote, and I've got them spelled out here. You can read about it. The next section heading should be history. How long have we known about the disease? Historically, how was this disease treated? What's the history of the vaccine or the development of the vaccine for this disease? And then describe the changes in our knowledge and attitude towards this disease throughout history. Even if you're doing a really recent disease like COVID-19, there has been a change in our knowledge and our attitude of the disease. And then a section on epidemiology, describe the prevalence and transmission of the disease. Where in the world is the disease prevalent? How many people are infected? What's the rate of new infections? What's the rate of morbidity and mortality? What is the reservoir of the pathogen? And what are the modes of transmission? Then the next section heading will be pathology. Describe the pathogenic effects on cells, tissues, and organ systems. What tissues, organ cells does the pathogen affect? What damage does the pathogen inflict? How is the damage inflicted? Is there direct mechanical damage? Is a toxin produced? Is the immune response causing the damage? What's the sequence of the disease? Like what's the length of incubation, the prodrome period, the period of illness? I guess we haven't talked about this, so we will, in which case you'll have a better idea. The period of decline, the convalescence. And then what are the major signs and symptoms of the disease? And then a section on response and treatment. I'll let you read about what you have to have on that section. And then a section on the social, political, economic considerations. And I'll let you read about that. I've got a grading rubric on the Canvas webpage. You can take a look at how the page will be graded. I've actually got it here too. The cover information will be 1 point. The causative agent will be 9 points. Like if it's a bacteria, you better put on the gram reaction, the shape, the cellular arrangement of the cells, and then whatever is important for that cell, like the metabolic capabilities, etc. Then for the history, it'll be worth 10 points. Talk about how long we've known about the disease, the history of vaccine development. And if there is no vaccine for that disease, just say there is no vaccine. History of the treatment of the disease, changes in our knowledge and attitudes of disease throughout history. And then for each of the other sections, I have what I'll be grading you on. Quality of writing will concern how good your grammar is, how good your spelling is, punctuation, how good the paper flows, how good it is or how easy it is to read, etc. And the quality of writing will be with 15 points. I've already mentioned the in-text citations worth 10 points. The full citation, the literature citation page will be worth 15 points for a total of 100 points. Any questions about any of that? So read about this. I think I've talked about everything, but let me look it over just to make certain. I do think maybe a student. Or say anything. I don't see, oh, I got the rubric attached to the instructions, that's right. All right, so this is just a Word document. Any questions about any of that? It looks like I do not have an example. And it's due February 18th. Okay, if there's no questions, that's it for the infectious disease project. It looks like 3 more people have sent in something, so I'll take a look at that after the lab. Oh, I never shut that down. Let's shut that down. All right, let's talk about the lab. We're talking about lab 2, aseptic transfers. Upon successful completion of this lab, students should be able to understand the purpose of using aseptic transfer techniques, determine if aseptic transfer was used from the data provided, and be able to define the following terms: ubiquitous, aseptic transfer, inoculation, and contaminant. There will be some video demonstrations in this lab. In our local environment, microorganisms are ubiquitous. This is a problem when working in a microbiology lab because we generally want to work with pure cultures, cultures that contain only one species of organism. Louis Pasteur developed the technique of using aseptic transfers to minimize the risk of culture contamination. Louis Pasteur also developed a technique for making a pure culture, but he used a liquid broth, and what he did was limiting dilution transfers. He would make a dilution, and if on only one test tube out of 10 test tubes had growth, he assumed that the one test tube was started by one cell or a few cells, so it was probably a pure culture. That was a very difficult way of getting a pure culture, but it was the first way developed to getting a pure culture. It was actually Dr. Koch who invented a much easier way to getting a pure culture, and we'll talk about that when the time is appropriate. We're also going to learn how to make an aseptic transfer in this lab. All right, during inoculation, you should never lay your test tube caps or petri dish lids on the workbench. If you do, they may become contaminated, and then when you put the cap on the tube, the contamination may fall into the tube. To minimize contamination when inoculating media, you should take off the cap, hold it in your hand, and then hold the tube at an angle, the uncapped tube at an angle, and that will reduce the chance that if anything falls down from the air, that it'll fall into the tube, because you're reducing the surface area exposed when you hold it at an angle. And for a plate, what you should do is take off the lid, but not take it off entirely. Have part of the lid cover the plate, and then streak on your plate. And then if something falls down from the air, it'll fall onto the top of the lid and not fall into the dish. It just will reduce your risk of airborne contamination. And then the greatest thing for reducing your risk of airborne contamination is to cap the tube or put the lid on the plate as soon as you're done with the plate. Meaning leave it exposed to the air for as little time as possible. In order order to inoculate, that is, transfer cells to a new sterile media, we must make sure that we sterilize everything that can potentially contaminate the sterile media. Obviously, before you start, you should wipe down your counter. We already mentioned that in the last lab. You should also use sterile media and then something to sterilize your tools. We would use a Bacto incinerator, which once you turn it on, it will become red hot in the stone in there, and then you sterilize the loop inside the Bacto incinerator. And there's a picture of a loop. You only put the wire portion of the loop in the Bacto incinerator. You should know the terms of this lab. You're not physically going to be performing any of these activities because the lab is all going to be done online, but you will be watching videos of some of these activities, and we expect that you will know and understand the material in this lab and in these videos. In procedure 1, you're going to transfer a sterile liquid using a glass serological pipette. In procedure 2, you're going to transfer a sterile paper disc into a tube using sterile forceps. So we have a link for you to watch a video demonstrating proper aseptic transfer. So watch that before you do the lab. Procedure 1, you're going to use aseptic technique to transfer 1 ml of sterile water into a nutrient broth. Now, you're not actually going to do this. You'll be watching a video about it. When you're transferring a liquid using something that's marked, you should note that water does not form on the top in a straight line. It forms a meniscus where it'll be this U-type shape. And how do you read the meniscus? Do you read it at the top, the bottom, or in the middle of the meniscus? For the meniscus, you actually read it at the bottom. And so when you're taking liquid out of the pipette, you read it at the bottom of the meniscus. If you're using a birette, which is what this looks like, you read it at the bottom of the meniscus. Water in particular forms a very sharp meniscus, and that's because water is a polar molecule. It is attracted to the polar glass. Any questions about any of that? So you'll take 1 ml of water and put it into a tube of media, and then incubate the tube of media for, I think, a day at 35 degrees in the incubator, and then take a look at it. For procedure 2, you're going to be aseptically transfer a sterile paper disc into a tube of nutrient broth. You can read the procedure there. Let me state how you do the aseptic transfer. For the pipette, obviously you use a sterile pipette and you use sterile water, and you take the pipette and take up 1 ml and then put it into your nutrient broth. For the aseptic transfer of the paper disc, you get a sterile paper disc, and it'll be covered with the lid to the petri dish, and you sterilize your forceps. Generally, to sterilize the forceps, you get them in alcohol. Generally, you put them in a beaker of alcohol, and then you flame the forceps, meaning while they're still wet with the alcohol, you put it up through the Bacto incinerator, and it'll cause the alcohol to catch on fire. Any question about any of that? And then you grab the sterile paper disc, taking only one, and then you put it into the sterile nutrient broth, and then quickly cap it. Incubate it overnight, and then take a look at the pictures for procedures 1 and 2. I'm going to move ahead to the procedures since I'm just talking about 1 and 2. So that's the sterile broth at the start. moving down to the pictures. This picture is procedure 1. This picture is for procedure 2. You can notice a difference here. With procedure 2, you can actually see the paper disc in the bottom of the tube. Any question about any of that? All right, I think we're on procedure 3 now. For procedure 3, you're going to be transferring Serratia marcescens to a tube of sterile broth. And you're going to be taking Serratia marcescens grown in a broth, I think. No, it's not grown in a broth. It's grown on an agar slant, or grown on a petri dish. So you take the cells and you put it into a liquid broth. And then in procedure 4, you're going to transfer Serratia marcescens and put it on an agar slant. For procedures 3 and 4, we have two video watching how to correctly transfer from a bacterial stock culture to a sterile broth and to a sterile slant. There is one note about the video, and that is Serratia marcescens is a bacteria which is red in color. So you can use that information in what grows up because of the color. And when they're using the video, the bacteria they're using in the video is off-white or white, so you can't have the color. For the video, you only need to watch to 4 minutes and 23 seconds. And the bacteria they're using is E. coli, so it's off-white. It's not colored red like Serratia marcescens. There's also an error in the video between 3 minutes and 5 seconds and 3 minutes and 20 seconds. Do you know what that arrow is? The hint is you can hear sizzling when he puts the loop into the liquid culture of E. coli. Or is it the solid culture of E. coli? You hear sizzling. And that was an error that the person demonstrating it made in the video. Any question about any of that? All right, so for procedure 3, you're using aseptic technique to transfer Serratia marcescens to a sterile broth. A broth culture is one that is liquid. Well, broth cultures are problematic because it's difficult to determine if minor contamination occurred because all the cells mix together in a broth. So you can only, in a broth culture, determine if there's major contamination. Meaning if 10% or more of the cells were, or 10% or less of the cells were contaminated, but 90% were Serratia marcescens, the broth would still look pinkish. You would not see the off-white color from the contamination in a broth. So my point is broth are a little tricky and you can't be certain that you don't have minor contamination. However, if you had major contamination, the growth will not look pinkish. It would be more of the off-white color with maybe a very slight pinkish color, but mostly off-white for major contamination. Any question about any of that? All right, let me remind you, when you're using a tube, you should have it properly labeled with your name, the date, the type of medium it is. In this lab, we're using NBYE, nutrient broth with yeast extract. We're going to be growing it at the incubation temperature of 30 degrees, which this is optional. You don't need to include that. And we're going to be growing Serratia marcescens or S. marcescens in this case. So you have to have the inoculum, the type of medium, the date, and your name. And then the temperature it's grown at is optional. And then we're going to put it in the incubator overnight at 30 degrees Celsius. For procedure 4, we're using the same bacteria, Serratia marcens, but we're going to be inoculating a sterile agar slant. And an agar slant is solid. Contamination can more easily be seen on a slant or a plate than in a broth, because if you even have only a little bit of contamination, you will note the coloration isn't correct in one area where you have the bacteria growing. Any question about any of that? So normally you'll streak it out something like that on the slant. And if it's all red, then there's no contamination. But if one area has a different color, like off-white, then you have a contaminant. You should also have this tube appropriately labeled. And we're going to grow this at 30 degrees overnight. Interpretation of the results. Online students are not going to be making these observations in a lab, but we will show you pictures and you have to interpret the pictures. You should be familiar with the possible outcomes and the associated terminology. You should use this table for interpreting your results for procedures 1 and 2 and the conclusions. For procedures 1 and 2, if the nutrient broth is clear, no organisms are growing in the broth is your conclusion, and aseptic transfer was successful. If the nutrient broth, on the other hand, is turbid, hazy, or cloudy, the conclusion is organisms are growing in the broth and aseptic transfer was unsuccessful. And you will be asked for what are the results for procedures 1 and 2 and what are the conclusions. And if you don't use the wording of the conclusion in this table, you will get it wrong, meaning you do need to state whether organisms are growing or not and whether aseptic transfer was successful or not. For procedure 3, if the nutrient broth is clear, no organisms are growing in the broth, and aseptic transfer was unsuccessful. Because if you remember in procedure 3, you are inoculating the broth with Serratia marcens. So we expect Serratia marcens to grow in the tube. If the nutrient broth is turbid, hazy, or cloudy, organisms are growing in the broth, and aseptic transfer was successful. For procedure 4, if the result is possible, if the slant does not show growth, colony or confluent growth on where you put the slant, then the conclusion is no organisms are growing on the slant and aseptic transfer was unsuccessful. If the slant does show confluent or colony growth, then organisms are growing on the slant and aseptic transfer was successful. And here are the pictures for the results. We give you pictures because you can't actually do this experiment. For procedure 1, there's the result. For procedure 2, there's the result. For procedure 3, here's the result. For procedure 4, there's the result. You note that when Serratia marcens is grown in a broth, it'll have a pink color. But if it's grown on a solid media like a slant, it will be a red color. And then describe the, work on the questions. Let me go to that because I think this is not correct. On your worksheet, it gives you the different things to view. And there's the table you should get in your results and conclusions from. I thought I had that on yours, that this is actually question 0. Describe the initial clarity of the nutrient broth prior to inoculating it with sterile water or the paper disc. And we did give you a picture of that. And that's up further in the manual. And then question 1 for procedure 1, describe the clarity of the nutrient broth after inoculating with sterile water. Was it clear or turbid? Based on these results, what conclusion can be drawn? Make sure your conclusions come from the table. And then list 3 possible sources of error with a septic transfer technique that could explain why the nutrient broth showed growth of microbes. And you do need 3 sources of error. For 2, describe the results of procedure 2. Describe the clarity of the nutrient broth after inoculating with a paper disc. Was it clear or turbid? Based on these results, what conclusion can be drawn? And then 3, describe the clarity of the nutrient broth after inoculating with Serratia marcescens and overnight incubation. Was it clear or turbid? And based on these results, what conclusion can be drawn? Once again, make sure you take the table for the conclusions. And how do you know whether you have transferred the correct microbe, Serratia marcescens, or not? There is a way to know. Result 4, is there bacterial growth on the slant? Yes or no? And how do you know? Just state why you know. Could you see bacteria growing on the slant or no bacteria growing on the slant? Does it appear to be the bacteria you inoculated or a contaminant? And that you should be able to answer because you use Serratia marcescens. All right, I think that's it for the lab. Go ahead and get started on the lab. If you have any questions, I will be here until 8 o'clock or the last student logs out. During last lab, everyone logged out very soon, and that's fine. If you do have a question and I'm not here because everyone's logged out, you'll have to email me. All right, go ahead and get started on the lab.
I did have a question about this particular lab we just talked about.
Okay,
I don't see a place to submit the answers to it. Is there a place to submit on the module?
there should be. Let me take a look. Should be under the worksheet, lab 2. Yeah, you should have a link down here, which is a submission link. I don't have a submission link because I'm an instructor. I've got a speed grader link.
Oh, I must have just looked right over it. Thank you very much.
Okay. It could be on the top right if it's not down on the bottom. All right, get started in the lab. I'm going to stop sharing my screen, but I will be here to answer questions.
question.
Mhm.
So for the last lab, you said we could do corrections. Do you want us to resubmit it on the same, on this where it says, where we submit our submission of the last one? Do you want us to do the correction on the same one, even though it's like late?
Oh,
Can we
it will be
resubmit it?
marked late, but that's only because Canvas does not have the capability of having two submissions. But don't
Okay,
worry about
perfect.
that. As long as you get it by the until date, it won't be late. But yes, just
Sounds good.
submit it the
Thank you.
same place you submitted the other.
Thank you so much.
No problem.